Chapter 8, Topic 3, What is DNA recombination? In this topic, we will talk about what DNA recombination is, horizontal gene transfer, conjugation, transformation, transduction, and transposons. There are just two objectives for this topic: be able to define recombinant, and to be able to describe three forms of horizontal gene transfer used in bacteria. So what is DNA recombination? There are two types: the naturally occurring process that you can see defined above, as well as the biotechnology application. The simplest version of these definitions is that through a variety of means, new DNA is inserted into the existing genome of an organism, altering the genomic code. Horizontal gene transfer is the most common way for bacteria to share their genomic information. It is called horizontal gene transfer because it comes from two adult cells and not from a parent cell. Genes passing from parent to offspring are known as vertical gene transfer, but we aren't going to talk about that here. There are three types of horizontal gene transfer: conjugation, transformation, and transduction. We are going to look at each of these in depth on the next several slides. Before we move on, we need to refresh our memory on how these processes occur. Each of them requires the transfer of genetic information either as a plasmid or as a DNA fragment. Plasmids are small circular segments of DNA that exist naturally in many bacterial or fungal organisms. An important aspect of plasmids is that they contain an origin of replication. This means that they do not have to fully integrate into the new cell's DNA to be read and to have gene product produced. Some plasmids will integrate, but it is not necessary. The DNA fragments have to be integrated into the DNA to be read, which makes their integration more difficult. Conjugation is sometimes referred to as bacterial sex because it is the only form of horizontal gene transfer where there is direct contact between the two cells. In conjugation, two live cells share genetic information via the pili or a specialized opening. This channel then allows DNA to be passed between the cells. This is the process where plasmids containing resistant genes are passed, creating drug-resistant bacteria. Your text goes into detail about how conjugation occurs, but I only want you to understand the basic process and the importance of this process. Transformation involves one live cell and naked DNA in the solution. This process is different than conjugation in two important ways. First is that the cells never come in contact. The second is that instead of a plasmid being exchanged, DNA fragments are released by the donor cell, either while it's living or after the donor cell dies, and the recipient cell takes up these fragments. An important aspect of this process is that the recipient cell must be competent. This means that it is capable of accepting the DNA as long fragments of naked DNA. Not all cells are competent all the time, so this is a very important requirement. One more note on this process is that because there is no direct contact, the genetic information can be passed between a variety of cells and species. This means that the genetic information can be spread to a variety of cells and not kept within a specific species of bacteria. Transduction occurs via bacteriophages. When we discussed these bacteriophages in Chapter 5, we talked about how they use bacterial cells to replicate themselves and spread. Transduction takes this process in a slightly different direction. In transduction, the bacteriophages mistakenly take up some of the host DNA along with their own. When they are released from the cell via lysis, they carry this DNA with them to the next cell. This information then gets incorporated into the new cell. However, this wouldn't matter if the cell was only going to be taken over by the phage. However, when the phage took up the bacterial DNA, this made the phage inactive and incapable of infecting new cells. This allows for the DNA to be transferred and incorporated into the new cell without it being killed by the phage. One last type of recombination we need to talk about are transposons. This occurs within one cell and is not necessarily transferred between cells. Transposons are sometimes called jumping genes. These genes can easily move through the genome of an organism. When these transposons move between genes in the genome, they can disrupt or alter the way the genes are read. This can turn genes off entirely, change the way they function, or turn on genes that weren't on before. This movement has been associated with changes in phenotypes and drug resistance. Well, that's it for this topic. As a reminder, here are the learning objectives for this topic. Be able to define recombinant, and be able to describe 3 forms of horizontal gene transfer used in bacteria.